Hi friends, welcome back to the session 3, Cell the Unit of Life. In session 2, I have already given you some of the questions to solve. You can see, check and verify your answers. Your first question was, name 2 cell organelles that are double membrane bound. What are the characteristics of these 2 organelles? State their functions and draw the labeled diagrams of both. See, different questions are combined together here in one. The first part, name 2 cell organelles that are double membrane bound. Yes, your answer, chloroplast and mitochondrion. Both are double membrane bound organelles present in the eukaryotic cell. Now, the second part, what are the characteristics of these 2 organelles? Characteristics, first one, both are double membrane bound, contain the fluid matrix and both will contain circular DNA. Both contains 70S ribosome, enzymes, ATP molecules and mineral ions. Yes, of course, both are self replicating organelles. Now, their difference is mitochondrion contains cristae, the enfoldings of the inner membrane, which increases the surface area. Chloroplast contains granum for light absorption. Now, regarding their functions, state their functions. Yes, of course, the function of chloroplast, the main function, help in photosynthesis. Already I have told you they can help in protein synthesis, they can also help in cytoplasmic inheritance, that means hereditary in function. Similarly, mitochondrion, you have already learned, helps in cellular respiration, ATP production, storage and distribution. Again, they can also synthesize some of the proteins, they are also hereditary in function. Your second question, it was, what are the characteristics of a prokaryotic cells? You check your answers. Small in size, nucleus absent, membrane bound organelles absent, nuclear membrane absent, the ribosome, that is 70S ribosome, they contain mesosomes, the enfoldings of plasma membrane. Spindle fibers are not formed during cell division, nucleolus is absent, both transcription and translation takes place simultaneously in the cytoplasm. Now, here is your third question. See your answers. Multicellular organisms have a division of labor. Explain in brief. In multicellular organisms, there is differentiation and specialization of cells in their structure, function and location. They are organized to form different tissues, which in turn form different organs for performing specific functions. Hence, division of labor increases the efficiency and survival in a multicellular organism. Now, your fourth question. Cell is the basic unit of life. Discuss in brief. See your answers. All the vital functions of life can be carried out in a single cell. For that purpose, each cell contains all the organelles for various vital functions, which itself shows division of labor in a prokaryotic cell or even in a single cell. Example, respiration is carried out by mitochondrion, digestion is carried out by lysosomes, reproduction by nucleus in a eukaryotic cell. Now, come to the important another organelle, lysosome. It was discovered by Christian D. Dewey in liver cells. It is a small vesicular structure seen in the cytoplasm, and it is meant for intracellular digestive activities. It is absent in prokaryotes and present in all the eukaryotic cells, animal cells, except in mammalian RBC. They are spherical in shape. They are seen in large numbers in cells that are involved in intracellular digestion, such as WBCs and phagocytes. It contains an outer single layered membrane similar to that of plasma membrane, made up of lipoprotein in composition. It contains a fluid substance, ground substance inside it, which is granular in nature and it is heterogeneous. And this particular granular heterogeneous ground substance present inside the lysosome, that is known as matrix. And this matrix, it contains acidic hydrolysis of pH 5, proteases meant for digestion of protein, nucleases, digestion of nucleic acids, glycosidases, phosphatase and esterases, etc. Now, come to the different types of lysosomes. Types of lysosomes or the different types of storage granules. There are different types. First one, primary lysosomes. They have digestive enzymes in inactive form. So, this particular lysosome, it is inactive in nature. Now, coming to the second one, that is known as secondary lysosome. It contains ingested food and the different digestive enzymes. Since it contains the ingested food and the digestive enzymes, it is known as heterophagosomes, or they are known as storage vacuoles. Now, the third one, that is known as autophagosomes. It is formed by the fusion of primary lysosome and the reserved food molecule. The fourth type of lysosome, known as residual body or tertiary lysosome. It contains only the undigested food which is later expelled out by exocytosis. Thus, lysosomes are called as polymorphic organelles, because polymorphic means, poly means many, they are seen in different forms. Now, coming to the functions of this lysosome. First one is heterophagy, digestion of foreign particles that are inviting into the cell. So, it is known as heterophagy. Second one, that is autophagy, which means digestion of the reserved food or some cell organelles during the time of starvation. The third one, autolysis. Auto means self, lysis means breakdown. Self digestion of cell. So, it is known as suicidal bags. Now, coming to endoplasmic reticulum. The term was given by Porter. It was discovered by Garnier, and its ultrastructure was given by Porter, Fulham and Clot. It is a network of interconnected cisternae, tubules and vesicles. They are present in the cytoplasm, mainly in the endoplasmic region. They are seen in the form of a network. So, it is known as a reticulum, and this reticulum or network like structures made up of cisternae, tubules and vesicles which are seen in the endoplasmic region of cytoplasm. Hence, it is known as endoplasmic reticulum. They are absent in all the prokaryotes just like other membrane bound organelles, and they are present in all the eukaryotes except the case of mature mammalian RBCs. Here you can see the diagram of an endoplasmic reticulum. See this structure, it consists of, it is a network of tubular structures and it is a highly branched one. Here you can see some of these spherical particles, we can call it as ribosome. So, this network like structure made up of different components. When we analyze this, we can see three distinct components and they are seen in the endoplasmic region of cytoplasm. So, the term endoplasmic reticulum. Now, the three different components are, you can see here cisternae, the second one that is tubules, third one vesicles. Now, see the structure and try to identify these three. Cisternae are tubular, flat, membrane bound, sac-like and unbranched. They are arranged in parallel groups or bundles. So, such structures, they are known as cisternae. Now, coming to the second one, that is tubules. They are tubular, elongated, thin, flat, but they are highly branched ones. And these branched structures, they will interconnect the cisternae with that of vesicles. The third one, that is vesicles, small spherical structures, they are known as vesicles, and cisternae and vesicles and tubules together forms the endoplasmic reticulum. Now, coming to the types of endoplasmic reticulum based on the presence or absence of ribosomes. Here the first category, that is smooth endoplasmic reticulum. Smooth endoplasmic reticulum, we are using the abbreviation SER. Ribosomes are totally absent on its surface. So, they are agranular endoplasmic reticulum. Here the granular substance, the granular particles, spherical particles, they are the ribosomes. So, if ribosomes are present on the surface of endoplasmic reticulum, then we will call it as rough endoplasmic reticulum. And if ribosomes are absent on the surface of endoplasmic reticulum, then we will call it as smooth endoplasmic reticulum. The second one, that is rough endoplasmic reticulum, we will call it as RER. Here you see this particular diagram, whether it is a rough endoplasmic reticulum or it is a smooth endoplasmic reticulum. Try to identify. Here the surface of this particular endoplasmic reticulum is rough in appearance because of the presence of granular spherical structures, that is the ribosome. So, since the ribosomes are present on its surface, this particular endoplasmic reticulum, that is a rough endoplasmic reticulum. Now coming to the ultrastructure, an endoplasmic reticulum is made up of 3 components. Already I have told you these 3 components. Here you can see again the cisternae, tubules and vesicles. You can see the characters also by minutely observing this diagram. Cisternae, they are narrow, tubular, membrane bound, unbranched, flat, sac like structures arranged in parallel groups. All these characters that you can observe in this particular diagram here. Similarly, for vesicles, they are small structures, spherical in shape or oval in structure, and they are the membrane bound, single membrane bound structures. Coming to tubules, tubular and highly branched, single membrane bound structures. Now coming to the different functions of endoplasmic reticulum. You can see the different functions. Since it is a tubular structure, it is helping in transport of materials within the cell, and it acts as a skeleton of the cell. That means it acts as an endoskeleton, cytoskeleton. Inside the cell, it can act as the supporting material. It provides mechanical support and contribute to the shape of the cell. It helps in cell division. It helps in the formation of cell plate in the case of cell division in plants, plant cells. You know the phragmoplast, that is formed by fragments of endoplasmic reticulum during the cell cytoplasmic division of a plant cell. And similarly, they helps in the formation of primary lysosome. These small vesicles, they get budded off and they produce as the primary lysosome. Synthesis of nuclear envelope during telophase. So, already I have told you they helps in cell plate formation. It is the mode of cytokinesis in the case of plant cell. Do you know by which method the cytokinesis takes place in an animal cell? Of course, it is cell furrowing, furrowing of the plasma membrane, divides the cytoplasm equally into 2. It helps in the storage of synthetic products like glycogen. Rough endoplasmic reticulum, it helps in protein synthesis. Do you know why it happens? Yes. You know which organelle is needed for protein synthesis? Of course, we have already covered this aspect. in the previous session. It is the ribosome that helps in protein synthesis. And in this way, here the rough endoplasmic reticulum, since it contains ribosomes, this rough endoplasmic reticulum helps in protein synthesis. Here in this particular diagram, here you can see the structure of a particular ribosome. Already it is covered in the previous session. Even then you can go through here. The ribosome, each ribosome, it has 2 subunits. Here it is 50S subunit and 30S subunit of 70S ribosome, the prokaryotic ribosome. Here it is 60S and 40S ribosome subunit. This is the subunit of 80S ribosome. Since the ribosomes are present here on its surface of rough endoplasmic reticulum, it helps in protein synthesis. Then rough endoplasmic reticulum, they forms transition vesicles which will carry the different materials to the Golgi bodies. So, from rough endoplasmic reticulum, materials are transported to Golgi bodies through these transition vesicles. Smooth endoplasmic reticulum, since they lack the ribosomes, they help in the synthesis of non-proteinaceous molecules, mainly the lipid molecules. They are involved in the synthesis of some of the hormones, mainly the steroid hormones. They helps in glycogenolysis. You know lysis, breakdown. Glycogenolysis, breakdown of glycogen into glucose, and this will happen in the liver cells. It is a mechanism to maintain the blood sugar level. Now, it helps in detoxification of harmful drugs. Now, coming to yet another organelle present in the eukaryotic cell, Golgi body. It was discovered by Camillo Golgi in the nervous cells of cats and barn owls. It is absent in prokaryotes, since it is also a membrane bound organelle. It is absent in prokaryotes, present in all the eukaryotes except in mature mammalian RBCs as well as sieve tube cells. Plant Golgi bodies, they are not well organized. They are seen scattered irregularly in the cytoplasm. So, we will call these plant Golgi bodies as dictyosomes. Golgi bodies, they are also made up of 3 elements here: cisternae, vacuoles and vesicles. Here you can see the diagram of a Golgi body. Membrane bound organelle, and these membrane bound organelles, single membrane bound organelle. Here also cisternae are present, then vesicle is here and some of the vacuoles, and they have the polarity. They have a concave surface as well as convex surface. Concave side is always directed towards cell membrane. That means it is facing outwards in a cell, and this particular phase, it is known as maturation phase or trans phase. The convex surface is seen towards nucleus of the cell. That means it is facing towards inside, and it is called the formative phase, or we can say it as cis phase. So, there are two phases. One is concave phase facing outwards, or maturation phase or trans phase. The other one is convex phase, surface facing towards nucleus or inside, which is known as formative phase or cis phase. Now, coming to the functions of Golgi bodies. They are involved in cell secretion. They are involved in the packaging of materials, collection, modification and packaging of different materials in the secretory vesicles, and they help in formation of cell plate also during the cytokinesis of plant cell. Then they help in the formation of acrosome in the sperm head region. So, acrosome present on the tip of the sperm head region, which is a modification of Golgi bodies which contains enzymes for penetrating the different layers of ovum such as corona radiata, zona pellucida and vitelline membrane. So, it plays an important role in penetration of this sperm into the ovum by forming the acrosome of the sperm. Now, the next function, that is formation of primary lysosome, which contains inactive enzymes, synthesis of glycoproteins and glycolipids. Now comes to the most important organelle, the center of the cell, that is the nucleus. It was discovered by Robert Brown in the root cells of orchid. It is seen in all the eukaryotic cells except in mature RBCs, mammalian RBCs, sieve tube cells, tracheids and vessels. Generally, they are seen at the center of a cell, but there are exceptions. In the case of a mature plant cell, they are seen in the periphery. Then what will be there at the center of a plant cell in a mature plant cell? Yes, you can see it is a large vacuole. Normally, a single cell consists of only one nucleus, but there are certain cases where a cell contains many nuclei. So, such a cell with many nuclei, that is known as a cenocyte. Shape and size of the nucleus, that can also vary depending upon the functional status of the cell. Nucleus is made up of DNA, RNA, lipids and proteins along with mineral ions. A nucleus has structurally 4 different components. You can see here the diagram. of a typical nucleus. Interphase nucleus with the outermost part. Again it is a double membrane bound organelle having two different membranes, outer membrane and inner membrane. Each membrane is again made up of lipoprotein in composition, just like that of plasma membrane. See, here in between these two membranes you can see a small narrow space, just like that of the mitochondrion as well as the chloroplast, perinuclear space. And here you can see in between, here and there are minute pores, and these pores we will call it as nuclear pore. And what is the need for this nuclear pore? You see, molecules are transported into the nucleus from the cytoplasm and as well as from nucleus to the cytoplasm. So, this transport of molecules that takes place through the nuclear pore. So, here nuclear membrane, or otherwise we can call it as nuclear envelope or karyolemma. Karyon means nucleus, lemma means layer or even membrane. So, karyolemma, it is double layered or bilayered, lipoproteinaceous, made up of lipid and protein. Again it is trilaminar, just like plasma membrane, and already I have told you in between the two membranes there is perinuclear space. There are minute pores present in the nuclear membrane, we will call it as nuclear pores, and its average size is 800 angstrom. It controls the transport of molecules into the nucleus as well as into the cytoplasm. It helps in pinocytosis as well as phagocytosis. Pinocytosis means cell drinking. If a fluid molecule is taken in, then we will call it as pinocytosis. Phagocytosis is known as cell eating. If it engulfs a solid molecule, then we will call it as phagocytosis, intake of solid molecule. The second component, that is nucleoplasm. Here in this diagram also you can see the dotted portion. See, these dots indicates the fluid matrix seen inside the nucleus, which is known as nucleoplasm or karyolymph. Lymph means fluid, karyon means nucleus. So, we can say it as karyolymph, the fluid content present inside the nucleus. It is colloidal in nature and it is a homogeneous ground substance. It contains sugars, enzymes, ribosomes, nucleotides, messenger RNA and transfer RNA molecules. It maintains the shape of the nucleus. It helps in the formation of spindle proteins during cell division, synthesis of RNA and ribosomal subunits. Now, coming to the third part, that is nucleolus. It is discovered by Fontana in skin cells of eel, and Bowman coined the term nucleolus. Here in this diagram you can see this spherical structure without any limiting membrane, and this structure, that is nucleolus. It has no limiting membrane. It is mainly formed of ribosomal RNA and non-histone proteins. It is a storehouse of ribosomal RNA. It plays an important role in spindle formation during cell division. Now, the fourth part, that is the chromatin reticulum. You can see here this thread-like structure, ultra thin, and it is seen in the form of a network made up of chromatin, so known as chromatin reticulum. They are darkly stained network of thread-like structure. When we are staining it with any of the stain, nuclear stain, DNA stain, that is fulgent stain for example, it was first reported by W. Fleming. Chromatin fibers, that is made up of DNA, RNA and protein. There are two types of chromatin, euchromatin and heterochromatin. The first one, euchromatin, or U means true, true chromatin. They are less coiled, they contains less DNA compared to heterochromatin. They are functionally active and takes less stain. The second one, that is heterochromatin. They are deeply coiled one and it takes more stain, so darkly stained. It contains more DNA and it is transcriptionally inactive. Now, what is meant by a chromosome? You have already learned this concept in your chapter 6 of NCERT textbook. So, you memorize during the cell division the chromatin fibers, the chromatin reticulum present in this interphase cell, which undergoes coiling and thereby condensation to form a thick and rod shaped structures. We will call them as chromosomes. So, we can see the chromosomes during division phase and chromatin reticulum in an interphase nucleus. The term chromosome was given by Waldeyer. Number of chromosome is specific in a particular species. For example, if it is a Drosophila or fruit fly, the diploid number is 8, 2n is equal to 8. If it is a frog, then it is 2n equals 26. You all know that in human beings 2n is equal to 46. Here I would like to ask one particular question. If there is 2n is equal to 47 by XXY condition, what is the disorder? Of course, it is Klinefelter syndrome. And if it reduces into 2n minus 1 x0 condition, then the disorder is Turner syndrome. Good. Then coming to apes, 2n is equal to 48. There is an organism Ascaris megalocephala where the 2n number goes to 2. So, in each and every organism the chromosome number is specific. A chromosome has two equal halves, and we will call these equal halves as chromatids, and they are attached together at a point, and this point is known as centromere or primary constriction. Each centromere has two disc shaped structures called as kinetochores. Each chromatid in turn is formed of two chromonemata threads, which is made up of DNA and protein. In some chromosomes, there is a secondary subterminal constriction. We call it as secondary constriction. Such chromosomes are known as SAT chromosomes, chromosomes with secondary constriction are known as SAT chromosomes or satellite chromosomes. Chromatin fiber is made up of chains of repeated units called as nucleosomes. You have already learned about this nucleosome and its structure. They are histone octamers surrounded by DNA in your chapter 6. Types of chromosomes based on centromere position. Here you can see the diagram of different types of chromosomes based on centromere position. The first one, here you see the centromere is exactly at the center. So, such a chromosome we will call it as metacentric chromosomes with two arms of equal size. The second one, that is submetacentric. Centromere is seen a little bit above the center. It results in the production of two unequal arms. One arm is longer one and the other is shorter one. Now, see the third case, that is acrocentric chromosomes. Still more, the position of a centromere is a little above, almost going towards tip, producing two unequal arms. The last one, that is telocentric, where you can see the position of centromere at the tip portion or terminal portion. So, in this way we can group these chromosomes into 4 different types based on the position of centromere. First one, metacentric. You can correlate with the diagram also here. Position of centromere exactly at the center, so that we will get two equal arms. The second one, that is submetacentric, where centromere is near the center or it is a little bit away from the center, result in the production of two unequal arms. Third one is acrocentric. Centromere is subterminal in position with two highly unequal arms. One is very short and one is very long. The last one, that is telocentric, where the centromere is terminal in position with one arm only. Now, coming to the functions of nucleus. It controls the synthesis of protein. It contains all the machinery for protein synthesis. It contains genetic information, as the DNA contains genes, the hereditary units. It helps in the formation of ribosomes needed for protein synthesis. it induces genetic variation and helps in evolution. It controls gene expression and gene regulation. Now coming to the exercise questions given in your NCERT textbook in this chapter. We can solve, see the answers, describe the structure of the following with the help of labeled diagrams. First one, centrosome, second one is nucleus. Yes, already we have covered these topics in the previous session. Even then, we can quickly go through these answers. Centrosome is formed of two darkly stained granules. They are known as diplosomes, since they are two in number, or the centrioles. They are surrounded by an amorphous cytoplasmic area called as centrosphere. Centrioles and centrosphere together known as centrosome. Structure of centriole already we have covered in the previous session. Have a quick flashback. They are hollow cylinders perpendicular to each other. Each centriole is microtubular structure made up of microtubules arranged in 9+0 manner. Each microtubule is a triplet which is formed of 3 subtubules A, B and C. A subtubule is formed of 13 parallel protofilaments. B and C subtubules are formed of 10 parallel protofilaments. The C subtubule of each microtubule is linked to A subtubule of adjacent microtubule by AC linker, and hence all the microtubules are tilted at 40°. At the center there is a proteinaceous hub. The A microtubule is connected with the central hub with the help of proteinaceous radial spokes. Now coming to the nucleus, already it is covered just now only, has 4 components: nuclear envelope with the nuclear pores, outer membrane and inner membrane, double layered one, nuclear pores helping in transport of molecules. They also help in pinocytosis and phagocytosis. Nucleoplasm with the different components, the fluid structure, heterogeneous sugars, enzymes, ribosomes, nucleotides, messenger RNA and transfer RNA. It maintains the shape of the nucleus. It helps in the formation of spindle proteins, synthesis of RNA and ribosomal units. Nucleolus discovered by Fontana in skin cells of eel. Then it has no limiting membrane and it is formed of ribosomal RNA, non-histone proteins. It is a storehouse of ribosomal RNA. It plays an important role in spindle fiber formation during cell division. Chromatin reticulum, network of thread-like structure made up of chromatin. What are nuclear pores? Already have listened the definition and structure, function. Centromere also, what is a centromere and how will you classify chromosomes based on centromere? We have already covered this. Both lysosomes and vacuoles are endomembrane structures, yet they differ in terms of their functions and comment about it. Functions of vacuoles: it contains cell sap that exerts turgor pressure and helps in cell growth, stores water and minerals and nutrients, it stores waste materials. Lysosomes, regarding its function: heterophagy, autophagy and autolysis. Now, here you can see the differences between a plant cell and animal cell. I think after attending this session, you are able to identify the differences between a plant cell and animal cell. Now, here are some of the questions for you to think and answer. Name the following suicidal bags: power plant of a cell, plant Golgi bodies, universal organelle, universal membrane and outer membrane of vacuole. Name 2 organelles that contain 70S ribosome. Name 2 organelles that contain circular DNA. Which ribosome is known as eukaryotic ribosome? Write the differences between euchromatin and heterochromatin. Which organelle helps in protein synthesis? Which part of the chloroplast is involved in dark reaction? What are the 2 different types of endoplasmic reticulum? Which plastids give red color to rose flowers? Name the plastids that give white colors to flowers and name any 4 organelles that contains ribosome.